Thank you very much for coming. It looks like the rate of people coming in have slowed, so I'm going to get started. As announcements for the community, the next Myco Talks is on the 28th of October. So please go to the University of Exeter website if you want to register. I'll put that into the chat. But it's a fantastic series of talks. They're free to register, so please do so if you're interested. Those are the fourth working Thursday of every month. The next one is the 28th of October. So with that, I will pass it off to Liz, who's going to introduce our first speaker. Our second speaker today is Mark Gresnigt, and he received his BSc from Saxion University of Applied Sciences in the Netherlands, and then his MSc and PhD from Radboud University, where he also performed some postdoctoral studies at the Radboud University Medical Center. He then received an Alexander von Humboldt Foundation postdoctoral fellow to work at the Leibniz-Institute for Natural Product Research and Infection Biology, Hyphae. And in 2020, he opened his own junior group, research group there. And so today he's going to talk about the work from his junior research group on Candida and serum proteins. So Mark.
Thank you very much for the very nice introduction. Yes, as you very nicely stated, my group is interested in Candida albicans and Candida species and how they adapt to the human host. And we first started to look at albumin as a protein that is very abundant in the human body. So what I would like you to appreciate about Candida species is that these are opportunistic fungal pathogens and they need to be highly dynamic. So for example, during commensalism, they need to compete with bacteria and sustain their persistence in the microbiome. But during infection, they actually have no competition with bacteria, but they have to compete suddenly with an immune system. And this requires very different adaptations to persist in these different niches. And we have the feeling that host proteins and adaptation to host proteins might play a role in this situation. So to illustrate also these big differences is that, for example, during commensalism, there is this different chemical and environmental stimuli. However, when you start to treat with cytostatic therapy and antibiotics that remove the beneficial microbiota, and you change, for example, the diet, this fungi can turn pathogenic and invade the host. And to stress these different environmental stresses, I have two different videos and why these adaptations are so important. So in this video, you see that, for example, macrophage is very easily clearing a lot of Candida cells. However, in other situations, it can also be that the fungus is proliferating very efficiently, and in this case, even triggering macrophage cell death and vigorously escaping this immune recognition. So we are interested in what drives these different adaptations to escape this. And looking at a picture of a systemic infection, we of course see that Candida albicans or other Candida species, they can originate from the intestine, travel through the bloodstream to then infect the vital organs, such as, for example, the kidney. And you have this classical feature of commensalism to pathogenicity. And during this journey, the fungus travels through the blood, which is a very rich environment in different host proteins, immune factors. And we have a feeling that there might be something happening when the fungus enters this niche. If you look at the blood and you take the plasma and you run this over an electrophoresis, you see this massive peak of albumin. This is highly abundant in blood. And if you plot this on the concentration of albumin on commensalism versus pathogenicity on this map, you see that the more the fungus invades, the more and more albumin it will encounter. So a little introduction on albumin. It's a highly multitasking protein. So you can consider it as a Swiss Army knife. So it maintains the oncotic pressure in our blood vessels. It is a major antioxidant. It is a molecular shuttle to transport a lot of important metabolites, but also medicine in our body. And if we look at the infection models that we use, is normally in blood, Candida would encounter 55 to 35 milligram per milliliter of albumin. But in almost all infection models, this is 0 milligram per milliliter, because we do not want to introduce serum because it's a key stimulating factor of filamentation. So my master's student at the time, Sophie Austermeier, she was very interested in this and started to perform experiments where you add kidney cells in a well and you infect them with Candida, and you add this dye to stain host cell necrosis. And she did this in the presence and absence of albumin. And what she noticed is that when adding albumin, you see that the cell death, the necrotic cell death, very nicely imaged here by these red dots, is significantly delayed. And then we, of course, started to wonder whether this might be some interesting interaction with the fungus. However, these preparations that you can buy online of albumin, they contain all kinds of preservatives. So to exclude that this was one of the factors driving this, we filtered the albumin over a 30 kilodalton separator, and then we saw that still the fraction larger than 30 kilodalton was the effective fraction. So there was no case of preservatives. And also what we saw is that this albumin needs to be constantly present during the infection. If you just pre-treat the host cells with albumin, there was no improved damage, but also if you just pre-treat the fungus, there's also no improved damage. So this reduced damage. So this was interesting. And then we started to look at the morphology. And of course, you can imagine that if you inhibit the filamentation of Candida albicans, that it cannot cause a sufficient damage. But we saw that in both cases, there were massive hyphae present, and they were invading the host cells. And recently, the group of Bernhard Huber here, also at the HKI, published this very nice paper on how the hyphae formation and the delivery of the toxin associated with Candida albicans hyphae is crucial for the host cell damage. And based on this, we thought, well, maybe this albumin is interacting with this toxin. So we took the recombinant toxin, and then we treat these artificial membranes in collaboration with a group of Julian Necklick in London. And what we see normally is that if canalizin is in the model, you get this permeabilization of the membrane, and the electrical current is disturbed. However, when we were treating these canalizin with albumin, we see that the electrical current is not being disturbed, and there is a constant resistance of the membrane. So then we tried to do this in a cell experiment. So we stimulated the kidney cells with the synthetic canalizin. And then we neutralized with 5 milligrams per milliliter of albumin. And we could see that we can really neutralize up to 50 micromolar of canalizin, which normally induces massive cell damage. And also to show that that this is albumin and canalizin, that this interaction is also playing a role with Candida, we just saturated our albumin with canalizin with 50 micromolar, because we knew that we could use this concentration to saturate it, and then add it to Candida albicans. And then we see that the damage induced by Candida albicans cannot be neutralized anymore. So interestingly, if you then go to a mouse model and you would take the synthetic canalizin and would inject it in the bloodstream, there's so much albumin present that it should not cause any phenotype. And this is actually some findings of the group of also Julian Necklick. So they injected different concentrations of canalizin IV in mice and observed practically no phenotype in the mouse whatsoever. And we hypothesized that this was because of the neutralization in the bloodstream. Of course, there's a lot of other proteins also present. So to show that it is really in the serum, the albumin neutralizing the damage induced by canalizin, we took serum, which is normally very nicely, significantly reducing Candida albicans induced damage, or canalizin induced damage in this case. And if you then deplete the serum from human serum albumin, you can see that this damage is almost completely restored. So very nicely confirming that albumin is the factor in the serum that neutralizes canalizin. So to pinpoint what is actually the interaction, we use this hydrophobic dye. So this dye is normally binding the albumin pocket that binds hydrophobic proteins. And the rationale behind this is that the candylysin peptide has to insert in this phospholipid bilayer. So it has some hydrophobic properties. And when we, for example, bound this dye to albumin, you get a very nice fluorescent signal. But if you should have candylysin there in this pocket bound by the hydrophobic interactions, this molecule is not fluorescent anymore. And this was indeed the case. So we could displace this fluorescent dye with candylysin, suggesting that it binds the hydrophobic sites of albumin. And then we did some in silico modeling using predicted candylysin structures and also the albumin structure that is publicly available. And then we see this very nice interaction here in this domain. And this also contains some hydrophobic amino acids. And very interestingly, a few reports already shown that also for other toxins, so bacterial toxins, in this case, toxins from Clostridioides difficile, also bind exactly to the same structure here in the albumin protein based on these hydrophobic interactions. So based on this, we made this model about how albumin is protecting during infection. So when Candida is in the tissue, it forms this hyphae, produces the candylysin, which causes, of course, inflammation. And during the inflammation, the vasculature is getting permeable and there's plasma leaking out. And then the albumin can enter the host tissue together with the rest of the plasma and then neutralize the candylysin, preventing host cell damage. However, of course, this is a very nice approach, but this still doesn't explain why Candida can still cause infection. So we were thinking, how can Candida maybe overcome this protection? And there's already a very nice study last year published in Bio showing that, for example, albumin can facilitate heme acquisition by Candida albicans and thereby stimulate its growth. And similarly, we have seen that albumin is also providing iron to Candida albicans, which you can chelate away with an iron chelator. So it can also very potently induce the Candida albicans growth. And I want to bring you back to this graph because normally we see that if we have albumin present during infection, you see this very nicely reduced damage, which is mediated by the neutralization of candylysin. But if you just expose Candida albicans to albumin before infection, you see actually this mildly increased damage. And we went further on this. So Sophia Hitzler, who started in my junior research group, she was very interested in seeing how widespread this phenotype is. So she started to look at damage in longer term infections. And she used all kinds of strains ranging from low damaging strains to from non-damaging strains to highly damaging strains like SC5314. And when adding albumin, we saw consistently that you get a very nice and significant reduction of the induced damage. And this is likely the damage mediated by candylysin. However, at later time points when we are adding albumin, we see actually that an opposite effect is happening. Suddenly, the damage is being increased by albumin, and even species or strains, better said, that normally do not cause damage to the kidney cells, suddenly are being capable of causing significant damage. And the interesting thing is, if you look, for example, at these strains in their phenotype, the highly damaging strains, of course, these are very nice hyphae producers that indeed release candylysin, but some of the non-damaging strains actually form this pseudo-hyphae phenotype, and then suddenly they can start to cause damage when albumin is there. So this idea that candylysin needs to be presented into the pockets by hyphae is, of course, very nice during early infection, but it seems that during later infection, this is not always needed. And based on this, we, of course, know also other Candida species that are not even capable of forming hyphae. So, for example, Candida garbata, this is one of the second most common causes of candidiasis, But if we look at in vitro and in vivo models, mostly we see that this fungus is practically avirulent or not causing damage. So in our lab, Marina Pekmezovic studied Candida garbata in the context of vaginal Candida infection. So you might ask, well, this is not inside the body, this is not in the bloodstream, so what is albumin doing there? So epithelial cells are, of course, actively secreting albumin, resulting in a level of about 5 milligrams per milliliter of albumin in the vaginal secrete. Plus, if there's an infection, as I already explained previously, the vasculature can permeabilize, and then, of course, you can get more albumin into the tissue. So when she started to look at the induction of damage by Candida garbata, so we saw, of course, that very nice early during infection, you see this very nice reduction, which is likely due to the neutralization of canalizin. But Candida garbata, which normally is not causing damage, is suddenly significantly augmented in its damage potential. If you then look, for example, at the mechanisms, is we see that Candida garbata suddenly very nicely is increasing its proliferation as well as its adhesion to host cells. And we were looking what is actually causing this. And one of the things that we saw is that if we put garbata with albumin in a growth curve assay, it cannot grow directly on albumin. So we accepted the fact that maybe the host cells are responsible for this increased damage potential. And if you put fluorescently labeled albumin on host cells, you see that they very nicely take up this albumin in vesicles. And also, if you block different kinds of uptake mechanisms in the host cells, you can completely neutralize this damage potential of Candida garbata. So the uptake of albumin by host cells is crucial. And of course, these host cells, they also have all kinds of proteases to process these proteins that they take up. And what we actually see is that when we, for example, neutralize protease activity with these protease inhibitors, you can completely neutralize the augmented damage potential, but also the growth induced by albumin in this setting. So to look then what were the genes in Candida garbata that are mediating this increased damage potential, Marina did a very nice mutant library screen where we looked at mutants that are attenuated in their potential to have their damage increased by human albumin. And here 4 mutants popped out. So the OST3, CDC19, HEP5, and OST6. And when growing these four knockouts in normal YPD medium, you can directly exclude these 3 mutants because they have a massive growth defect in YPD by themselves. However, HEP5 normally grows like wild type. And if you then expose the HEP5 knockout to albumin, you see that you practically cannot augment its damage potential. And as you might know, HEP5 is a crucial regulator of iron homeostasis in Candida garbata. So we started to look at different aspects of iron acquisition. So when we, for example, chelated iron with BPS or with transferrin, which can also take away iron from Candida garbata, we saw that you can completely neutralize this increased damage potential. Also, on the contrary, when you, instead of albumin, add increasing concentrations of ferrous sulfate, you can very nicely increase the damage potential to the same extent as that you can do with albumin. So this hinted to us towards that maybe albumin is somehow, when it's processed by the host cells, delivering iron to the Candida garbata cells. But then we got a question like, how does Candida garbata cause damage? Because it has no toxin, it cannot form hyphae. So we looked at the transcriptional responses induced by albumin. And one very interesting aspect is that you get a lot of increase in glucose metabolism. And when you then are, for example, looking at the cytotoxicity, you see, of course, that the glucose concentration in these experiments is very nicely depleted. But when you are supplementing with additional glucose, we can significantly reduce this damage potential by Candida glabrata. So glucose starvation is definitely one of the host aspects that is causing the damage. But if you do experiments, for example, in medium without glucose, you can practically not see any host damage at all. So it has to be a combination of glucose consumption and another mechanism. So we also transcriptionally profiled the host response to Candida glabrata in the presence of albumin. And here, very interestingly, we found that the ER stress response is being induced. So we have the feeling that the combination of glucose consumption and an ER stress response is responsible for this increased damage in the presence of albumin. So Marina made this very nice model where we see that if the epithelial cells are taking up albumin via macropinocytosis, they proteolytically degrade the albumin. They deliver these peptides that are derived to the fungus, which of course triggers Hep5 dependent transcriptional responses, which leads to an increased epithelial cell damage due to glucose starvation and ER stress. But now you might think that here's a problem, because there's normally not damage being induced by Candida glabrata, and there's always about 5 milligrams per ml albumin present in the vaginal secrete. So how is this relevant? So then we had a look at what is actually normally keeping Candida infections at bay. And of course, the vaginal microbiota plays a crucial role. And also in the group of Bernard Huber, we have also observed and shown that you can keep Candida species, and in this case, it was Candida albicans, commensal by supplying lactobacilli and colonizing the epithelium with lactobacilli. So we tested the same with Candida glabrata, and what we see is that if we colonize the epithelium with lactobacilli, the albumin cannot increase the damage potential of Candida glabrata anymore. So now we can say that albumin is only augmenting the damage potential of Candida glabrata when this protective microbiota is disturbed, which of course fits the predisposing factors for vulvovaginal candidiasis, like for example, the use of antibiotics. So and with that, I come to my conclusion. So what we've seen is that during infection, Candida species are encountering host proteins that are often disregarded in infection models, but we see that these proteins can have a very important impact and a very strong influence on the outcome of infection. We've seen that albumin can neutralize Candida lysin, but at the same time also increase pathogenicity of Candida species. And we believe that these fungal responses to albumin can also help to explain why some of the clinical isolates that we get from patients that when we use them in our lab can practically not cause any damage to host cells. And we think that maybe albumin is playing a big role during infection with these strains. So I would like to acknowledge, of course, my collaborators, and in particular the PhD students that did the work, Sophie Austermeier, Sophia Hitzler, and Marina Pekmesovic, and I'm open for questions.
Great, Mark. Thanks for another fantastic talk. And thanks to both our speakers. I think that those were both excellent talks and on very interesting data. All right, Mark, I'll start with a two-part question from Johannes Westman. And the first part is the inhibitory effect of albumin on Candida lysin specific, or does it also apply to other cationic amphipathic alpha helical peptides?
That is, of course, a very interesting question. And we asked ourselves the same question when we were working on the project, because we saw that these hydrophobic interactions play a big role. And of course, hydrophobicity is very general. So we practically took every hydrophobic toxin that we could get our hands on in a short time. For example, we looked at bee venom, which is very similar to Candida lysin, and this gets neutralized. But also, for example, bigger toxins like streptolysin O gets neutralized. But this is also a pore-forming toxin. So we looked also at staurosporin, which induces apoptosis. This gets neutralized. We saw that alpha-amanitin gets neutralized. So far, we haven't seen a hydrophobic toxin that doesn't get neutralized by albumin. So it could be that albumin is also somehow protecting us against intoxication.
And the second part of that question is, what is the albumin concentration in inflammatory tissue or other areas of albicans invasion?
Yeah, so in inflammatory tissue, it's of course very, very difficult to say. In the oral cavity, in the saliva, there's a lot of albumin being present. The exact concentration I do not know by heart because I did not look it up or we did not measure it yet. In the vaginal secrete, about 5 milligrams per milliliter is being reported. So definitely the concentrations of around 1 to 5 milligrams per milliliter, which we also use in our experiments, are being found. In plasma, it's of course much higher, 35 milligrams per ml, which is really a lot of protein.
Mm-hmm. And then Felipe Santiago Tirado asks about mucosal surfaces and sort of what are the differences in disease manifestation that could be attributed to these tissues having different albumin concentrations.
Yeah, here it's also very interesting. So we saw a big difference in our model between the kidney cells and the epithelial cells. So on the epithelial cells, we see that what I showed is this uptake of albumin. This is much reduced in organ tissue like the kidney cells that we use. I do not know whether this is for each organ tissue the same. But we see that this uptake and processing of albumin definitely plays a role. And yeah, I also think that the local concentration of albumin plays a role, but we should more or less maybe make a map of the different albumin concentrations in different tissues during infection, also during homeostasis.
Great. The next question is from Johannes Westman, and it says, Mark, can you expand more on the glucose data? Did the C. glabrata consume glucose directly in the culture media, or did the glucose take a route into the host cell? And if it's the latter, is it via glut transporters or via endocytosis?
Yeah, that's a very, very good question. We don't know actually. We see, of course, that if we do not supply glucose at all, we do not get the expansion of the glabrata population. And yeah, it's definitely an interesting idea to see whether it's stealing the glucose from the host cell, but we haven't looked at it yet, but it's definitely worthwhile to do.
Mm-hmm. And then Paul Fidel says, nice work, Mark. You're correct. The glabrata is not very pathogenic in animal models, including the vaginal model. Interesting though, the mouse doesn't harbor much lactobacilli. So intuitively, one might think glabrata would be more pathogenic in the mouse model with less lacto present. Do you know how much albumin is being produced in the epithelial cells in the mouse vagina?
No, we do not know how much albumin is being produced there. However, what we've seen in our experiments, and this is something we're still following up on, is that when we are taking human albumin, we could boost the pathogenicity of Candida glabrata. We were unable to do this with mouse albumin, however, but we do not know whether this is maybe because the host cells cannot take up the mouse albumin and process it, or that just the mouse albumin is not functional. But yeah, we're diving into this right now.
Okay, and then a moderator privilege, I get to ask a question, which is, could you exploit the albumin responsiveness of glabrata to predict infection severity? And does the cell surface differ between the albumin responsive strains and the non-responsive strains?
Yeah, so this is actually something that we are very interested and keen to work on. So we are trying now to get samples from VVC patients and preferably also healthy controls to correlate maybe albumin levels to disease severity or maybe inflammatory responses, which of course are a key hallmark of the disease. And the cell surface of the responsive strains. So far, we only have found responsive strains except for Candida parapsilosis, which is not responsive to albumin. But we are looking hard now for Candida albicans isolates that are not responsive. For the, I think with the surface, you also mean the knockout strains that we tested. But these, yeah, the 4 that did not respond drastically, they really have a growth defect except for Hyphae. So no, not really a surface defect.
Great. There's one last question that just came in from Mark from the chat, so I can do that one.
Okay.
Which is from Johannes Vestman, and it says, is albumin small enough to enter the Candida albicans invasion pocket in epithelial cells from the culture media? For example, he says, I think that IgG is too large to enter the invasion pocket.
So we have the feeling that it's not, and this is also one of the reasons why we see like still a gradual increase of the damage induction in our live cell microscopy. So we think that most of the canalizin that's being secreted in the environment by the hyphae on the surface of the cells is all neutralized, but the canalizin in the pocket, we definitely have a feeling that it's not being neutralized. Also, when we allow Candida albicans to invade first a little bit and then add the albumin later, we see that the damage is significantly higher than when you directly have albumin present. So we think that it's not getting into the pocket, but this is of course also worthwhile to check with microscopy.
I think we've gotten through all the questions for Mark. If you have a last minute question, please feel free to turn on your microphone. Otherwise, we will end the meeting for today with thanks to both our fantastic speakers. Those were great talks.
Thank you. Thank you so much, guys.
Yeah.
Wonderful job.